Well, good morning, everybody. Friday morning, 11 a.m., at least here in Roanoke, Virginia, so that can only mean it's time for the Fralin Biomedical Research Institute Pioneers in Biomedical Research Seminar Series. Welcome to everyone. Before I have the pleasure of introducing today's speaker, I just want to put in a reminder for next week's talk. Next week, same time, we'll have a presentation by Dr. John Thiefelt from the University of Kansas, where he is a professor in the Department of Molecular and Integrative Physiology. And Dr. Thiefelt will be talking about aerobic fitness and susceptibility to metabolic dysfunction. So I hope to see everybody back next week at the same time. So I'd like to get started and introduce today's speaker. It's a great pleasure to be able to welcome Dr. Roshan. Dr. Kuhls is professor and chair of cognitive neuropsychiatry in the Department of Psychiatry at Radboud University and Nijmegan Medical Center in the Netherlands. Dr. Kuhls also serves as the principal investigator at the Donders Institute for Brain, Cognition, and Behavior at Radboud University. She did her early studies at Groningen and then went on to do her MPhil and Ph.D. at Cambridge in experimental psychology, working with Trevor Robbins. She did a research fellowship at Cambridge and then moved over to Berkeley, UC Berkeley, where she did postdoctoral training in the lab of Mark Disposito. And she then obtained a Royal Society University Fellowship at Cambridge, and at that point in time took up her positions at the Donders and at Radboud University. Dr. Kuhls is extremely well known for her contributions to studying the chemistry of the adaptive mind, particularly motivational and cognitive control of human behavior, and how it's modulated by the major neuromodulatory pathways, particularly the ascending dopaminergic and serotonergic systems. She's been recognized for her contributions in many ways. Just a couple examples. She's an elected member of multiple prestigious scientific associations, including the Royal Netherlands Academy of Arts and Sciences, the International Neuropsychological Society, the Academy Europa, and the Association for Psychological Sciences. She also currently serves as a lead scientist in the Human Brain Project, and she's a recipient of the Emoto Science Award in the Social Sciences. Dr. Kuhls serves as chair currently of the international jury for the Carvalho-Heineken Prize for Cognitive Sciences and is a member of the Scientific Advisory Council of the Allen Institute for Brain Science. She serves on the advisory board of TINS, Trends in Neuroscience, and is a reviewing editor for the Journal of Neuroscience, and as well as editor for Computational Neuroscience. Dr. Kuhls' work, as I said, has made major contributions in understanding the importance and role of neuromodulatory pathways and behavior. Amongst those, just a few included her findings and demonstration that dopamine can both enhance and undermine human cognitive control. Studying the effects of dopaminergic drugs can be predicted from baseline levels of dopamine. And I'm sure we'll hear about the measures with dopamine PET or positron emission tomography. Also, the effects of dopaminergic drugs depending on the targeted region, and that dopaminergic compounds can modulate cognitive control by changing willingness rather than ability to exert control, as well as studying the effects of serotonin potentiating and inhibition and anticipating adverse events. So today we're going to hear about some of the role of the major ascending neuromodulatory systems from the midbrain in dynamic regulation of flexibility and stability and computational trade-offs facing a constantly changing task demand. And she will share with us her evidence from pharmacological PET, fMRI studies on cost-benefit decision-making about cognitive control. So without further ado, it's my great pleasure to welcome Dr. Roshan Kuhls. Welcome, Dr. Kuhls.
Thank you so much for this extensive introduction. Wonderful to be here. Can you now all see my slides?
Not yet.
Or do I need to, let's see.
You need to share here.
I have.
There you go.
Do you see my presentation screen or my full screen?
You have to change it over to.
We see the correct screen.
Okay, good.
Yeah, you see the full screen or the one with the notes?
Full screen.
Full screen, great, fantastic. Yeah, thanks for inviting me, introducing me to give this seminar. I would have loved, of course, to to be present, but alas, we adapt flexibly. And this is also the topic of my talk today, as Mike already told you. Demands for mental cognitive flexibility are probably higher now than they've ever been, in part because of the enormous uncertainty, volatility, and lack of controllability of today's environment. Now, I want to begin by acknowledging my wonderful team of students and postdocs and assistants and collaborators, and the fact that the work that I'll present today is the result of a major collaborative effort. I'll focus in the beginning of my talk on a study done primarily by Ruben van den Bos, a PhD student in the lab, and then move on swiftly to a project carried out primarily also in collaboration with Ruben, but with Andrew Westbrook, who was a postdoc in my lab, currently in Michael Frank's lab, and that study was in collaboration with Michael. And then I'll end with work done in collaboration with Romain Liol and Verena Lee. Romain was also a postdoc in my lab and currently in Portugal, working with Zach Minen. So I think it's fair to say that we live in the age of the connectome, where the brain is conceptualized as a distributed computing system consisting of multiple structurally densely interconnected neural networks that can now be characterized exquisitely well. But a key point is that these structural connections are relatively fixed in the adult brain. And so a major question is therefore, what are the mechanisms that allow our brain to adapt flexibly, which we do. Sometimes it's difficult, but we do flexibly adapt to the changes in our environment. And the take-home message of this talk is that the major ascending neuromodulatory systems that originate from the midbrain, in the brainstem, like dopamine, but also noradrenaline, serotonin, they're particularly well suited for this. So it's the chemical neuromodulators that adapt the output of our structurally fixed neural networks to our changing environment. So indeed, the brain constantly faces a variety of computational dilemmas, depending on task demands of our current environment. These neural networks need to exhibit stability or flexibility, instrumental or Pavlovian control, or they need to exhibit speed or rather accuracy. And these dilemmas are at the core of this construct of my interest and that of many others of cognitive control, which is an ill-defined construct, but it generally refers to those mental processes that allow us to obtain our goals by resisting distractions, temptations, and impulses. Cognitive controls are considered a hallmark of the human brain. And so I expect it won't be a problem for you whatsoever to focus on my video or this meeting for the next hour or so without being distracted, right? It won't be a problem at all. You have all very large prefrontal cortexes. The question is whether you will decide to make use of that wonderful capacity. After all, cognitive control is costly. It's intrinsically effortful, and it's also associated with missed opportunities, right? Sometimes adaptive behavior requires leisure, letting go of control, rather than labor and focus. And so really what we need is a meta-level ability, a meta-level control to decide when to exert cognitive focus and effort, and when rather to let go of focus and effort and flexibly respond to new input. How do we do that? Answering that question requires that the problem of cognitive control is reconceptualized as a problem of reinforcement learning and decision-making, instead of simply a problem of implementation and say, distractor resistance. And so this take-home message that the chemical neuromodulators like dopamine adapt the output of our neural networks is grounded in part in this observation that they contribute to arbitrating or decision-making between different computational strategies, often opponent computational strategies. But let me start with defining chemical neuromodulation. It refers to that set of neurochemical processes that curtail or prolong or augment or diminish effects of fast signaling in neuronal networks, often conditional on the baseline state of the postsynaptic cell. So this modulatory mode is often distinguished from fast direct signaling in the classic neurotransmitter mode via glutamate and GABA. And it's rather the control exerted over such classic neurotransmission. I'll focus on the dopamine system, as I said, which is so widely implicated in both healthy and disordered cognitive functions. Indeed, the cognitive role of dopamine is keenly illustrated, of course, by its implication in ADHD, Parkinson's disease, schizophrenia, drug addictions, but also healthy states of stress and fatigue. They're all accompanied, these states and disorders, by cognitive or impulse control deficits, which can in turn be treated with drugs that alter dopamine transmission. Unfortunately, there's huge variability in the effects, both direction and the extent of these drugs, both within individuals as well as across different individuals. For example, consider ADHD, where symptoms of inattention and impulsivity often treated with dopamine-enhancing drugs like Ritalin, methylphenidate, which is the active ingredient of Ritalin or Concerta. And conversely, consider Parkinson's disease, where dopamine-enhancing medication can actually, in a considerable proportion of patients, contribute to cognitive deficits, impulse control deficits, leading in some cases to severe psychiatric abnormalities, including behavioral addictions, pathological gambling, and drug addictions. So one of the key questions that drives much of our work is, how can it be that the same neuromodulator has such different effects in different individuals? And so let me illustrate how we have begun to address these questions in the past. Indeed, my PhD work, which I did with Trevor Robbins years ago, started with this dopamine overdose hypothesis, which was put forward to account for paradoxical effects of dopaminergic medication on cognitive functions in Parkinson's disease. So while medication is well known to remedy some forms of mental rigidity and, of course, motor deficits in PD, Parkinson's disease, it is also well known to contribute to these compulsive behaviors and impulse control deficits, as I just said. And the essence of the dopamine overdose hypothesis is that the anti-Parkinson medication doses that are necessary to remedy the severe lack of dopamine in the dorsal striatum, leading to impairments in circuitry connecting this dorsal striatum with dorsal parts of the frontal cortex. Those doses are actually detrimental, impairing. They can those same doses can detrimentally overdose dopamine levels in relatively intact parts of the brain, such as the ventral striatum, the limbic parts of the striatum, and strongly connected orbitofrontal cortex. So improving dorsal frontostriatal function on the one hand, while at the same time impairing ventral frontostriatal function. So at the time, we provided evidence for this hypothesis by showing first that patients on medication perform significantly better than patients off their medication on a tacit switching paradigm associated with dorsal frontostriatal circuitry, while performing also significantly more poorly than patients off medication on a probabilistic reinforcement learning paradigm associated with relatively intact ventral striatal circuitry. So this evidence constituted a double dissociation, strengthening this dopamine overdose hypothesis. Now, these data were grounded in evidence mostly coming from work with experimental animals, showing that there is an inverted U-shaped relationship between dopamine and prefrontal function. And what we did with this Parkinson work is suggest that that idea can be extended to striatal reinforcement learning, so that just like there is an optimal level of dopamine for prefrontal functioning, there is also an optimal level of striatal dopamine for reinforcement learning. Now, to test this baseline dependency hypothesis for striatal learning more directly, we've recently studied the effects of the most commonly used dopaminergic drug, namely methylphenidate, and leveraged large variation in baseline levels of striatal dopamine, this time in a large group of healthy individuals. So we've made use of the fact that there is a lot of individual differences in baseline levels of striatal dopamine, even in the healthy population. Indeed, methylphenidate has long been known to boost flexible cognition. And indeed, it's increasingly used for enhancing intelligence, concentration, focus, even in healthy people. So for example, a 2017 study showed that already 30% of Americans at that time had taken some form of smart drug like methylphenidate in the last 12 months, which was up from 20% in 2015. And this trend is continuing. And the aim of this study that I want to tell you about was to establish that methylphenidate's effects on cognition indeed reflect changes in striatal dopamine rather than, say, noradrenaline, and that variability in striatal dopamine levels can account for individual differences in the drug's cognitive effects. So to directly measure striatal dopamine, our subjects underwent an F-DOPA PET scan to quantify dopamine synthesis capacity. And basically, subjects would undergo a PET scan after injection of a radio tracer, in this case F-DOPA, and uptake of that radio tracer indexes the degree to which dopamine is synthesized in the striatal terminals of midbrain dopamine neurons. And it provides a relatively stable trait index of dopamine synthesis capacity. So here are just illustrating the fact that there's quite some variability in even healthy students. So this is an actual slice through the basal ganglia with brighter colors representing higher uptake of this tracer, indexing higher dopamine synthesis capacity. Now, in addition to undergoing this F-DOPA PET scan in this study, all subjects came to the lab for another 3 visits, during which they received one of 3 pills. On one occasion, they received 20 milligrams of methylphenidate and the ingredient of Ritalin and Concerta, or on a separate visit, 400 milligrams of sulpiride, which is a dopamine D2 receptor agent known to increase dopamine levels selectively at low doses, at this dose administered here, and of course a placebo session. And the comparison of the effects of methylphenidate with those of the selective dopamine agent sulpiride allowed us to further substantiate the role of striatal dopamine in methylphenidate's effects. And during these drug visits, so there's three drug visits, in addition to a separate visit for a PET scan, all subjects were scanned with fMRI, functional magnetic resonance imaging, while they performed a reinforcement learning task, a reward and punishment prediction task, in which a subject had to learn associations between stimuli, in this case, faces or scenes, and rewards and punishments. And occasionally we would present unexpected rewards and unexpected punishment, which then signaled that things had changed, contingencies had changed, and subject had to update their predictions and change behavior. Now, the first striking finding of this large study was that both dopamine drugs, so both methylphenidate and sulpiride, increased bold signal measured with fMRI selectively in the basal ganglia, in the striatum, at the time of those prediction errors. So when people received an unexpected reward and an unexpected punishment, signal was significantly enhanced in the striatum, and that effect was boosted by these dopamine drugs. So in this slide here, I'm illustrating the effect of methylphenidate, but the same effect was seen for sulpiride. And what was more was that these drug effects on striatal bold signaling depended on baseline levels of striatal dopamine measured with PET. So here's the same picture as I showed on the previous slide, and this time stratified by individual differences in striatal dopamine synthesis capacity. So what you can see is that this effect of the drug is much greater, this boosting effect of the drug is much greater in subjects with low levels of striatal dopamine than in subjects with high levels of striatal dopamine. Really consistent with that baseline dependency principle. This was true for methylphenidate, but also for sulpiride. Interestingly, striatal dopamine synthesis capacity measured with FDOPA PET also predicted the behavioral effects of the drugs on learning. And so what I'm showing you here in these scans, in these images, are actually the results from the PET analyses. So this is not bold signal, but it's rather indexing dopamine synthesis capacity, such that signal corresponds to correlations between dopamine synthesis capacity and behavioral drug effects. Now, the precise pattern of this effect is complex, but best understood as reflecting greater boosting of punishment-based reversal learning in people with lower levels of baseline striatal dopamine, lower baseline levels of striatal dopamine. So together, these findings really substantiated the importance of striatal dopamine in methylphenidate's effects on flexible cognition, and also demonstrate that individual differences in striatal dopamine, in baseline levels of striatal dopamine, can account for variability in the cognitive efficacy of methylphenidate. But this study also allowed us to address a more specific question about the mechanisms by which striatal dopamine gates flexible cognition. Specifically, dopamine is perfectly placed, given the anatomical architecture of the striatal terminals of dopamine neurons and its physiological prediction error signature, to dynamically gate cortical representations depending on their task relevance. So consider a task context where subjects have to shift attention between faces and scenes, for example. And by analogy, consider the task under study here, where people have to flexibly update outcome predictions for either faces or scenes, depending on their experience and their relevance. That's based on prediction error. Now, what we see here is that such outcome prediction errors significantly enhance stimulus selectivity, plotted here on the y-axis, in visual association cortex. So in other words, to take you through this finding step by step, what we find here is that activity in the fusiform face area, which we know is specialized for representing faces, that activity in this FFA is disproportionately enhanced when a face is unexpectedly paired with an unexpected outcome, so requiring attentional reorienting so that new learning can take place. Conversely, activity in the parahippocampal place area, the PPA, specialized for processing scenes, is particularly enhanced when a scene is unexpectedly paired with an unexpected outcome. And that result is interesting in and of itself because it demonstrates prediction error-based, experience-based modulation of selective visual attention for learning. But what was even more interesting to us was that both drugs, both dopamine drugs, both methylphenidate and sulpiride, boosted this enhancement of stimulus selectivity, again in a manner that depended on baseline levels of striatal dopamine. So that the dopamine drugs boosted experience-based enhancement of selective visual attention in low dopamine subjects, but not in high dopamine subjects. So this work indicates that methylphenidate acts on dopamine in the striatum to modulate reinforcement learning and associated neural gating signals. And it also firmly establishes the baseline dependency principle for the most commonly used dopamine drug, methylphenidate. And an important implication of this is that isolation of dopamine's effects on cognition requires that one takes into account individual differences in the baseline state of the system. Now, and more specifically, our data also substantiated the hypothesis that striatal dopamine boosts experience-dependent dynamic gating of visual attention to currently task-relevant representations. This is something that I also said 2 slides ago. Now, this finding generally concurs with modeling work, for example, by Randy O'Reilly and Michael Frank, according to which dopamine opens the gate to cortex in proportion to prediction errors by having opposite effects on go and no-go pathways of the basal ganglia, boosting go pathway activity, but suppressing no-go pathway activity. The next question we asked is whether this gating role of striatal dopamine also extends to higher order goals, thereby affecting also the selection, the gating, the selection of entire cognitive tasks or task strategies, if you will. And we also asked, if that is the case, how does striatal dopamine then set the threshold for such high-level cognitive task gating? And according to currently popular expected value models of cognitive control, we allocate resources to a cognitive task based on its expected value. That's based on the benefits that it confers versus its costs. Specifically, we've been working with the hypothesis that striatal dopamine affects cognitive control task performance indirectly by modulating the value that we assign to cognitive work, namely by increasing the weight on the benefits versus the costs of a cognitive task by altering this balance between activity in a go and a no-go pathway of the basal ganglia. The general implication of this specific hypothesis is that the cognitive effects of dopamine-related disorders, like ADHD, schizophrenia, Parkinson's disease, and also the cognitive effects of their treatment might well reflect modulation of the willingness rather than the ability to exert control. And so to test that hypothesis with Andrew Westbrook, who was a postdoc in my lab at the time, and with Michael Frank, we have employed a cognitive effort discounting paradigm that Andrew actually developed when he was a PhD student in Todd Braver's lab. And that paradigm was used to assess what monetary value people assign to repeating a difficult cognitive task. And that experiment consisted of 3 different phases. In a first phase, subjects were exposed to a set of easy or difficult working memory tasks. So a 1-back, a 2-back, a 3-back, or a 4-back working memory task in which people were looking sequentially at letters, and they had to indicate whether the letter that was currently on the screen was the same as the letter just previously on the screen, or actually a few trials back. And clearly, the higher the N back, the more difficult the task. They were simply exposed to these tasks. Now, in the key phase, the second phase of the experiment, subject had to indicate with the button press whether they preferred to repeat after the test session was over, the easy or the difficult one of the tasks, difficult task, in return for more or less monetary payoff. And critically, the monetary payoffs for the easy task were titrated depending on choice, so that an indifference point could be derived, which basically corresponds to the subjective value that people assign to the difficult task. Now, in a separate session, participants underwent this F-DOPA PET scan. In fact, this study this experiment was part of the same overarching study that I described in the beginning of my talk. So subjects performed 1 PET scan, and then on the drug sessions, they performed multiple experiments. The reversal paradigm I explained, but also this cognitive effort discounting paradigm. So all subjects who were part of this experiment also underwent this PET scan to quantify baseline levels of striatal dopamine. And so that allowed us to assess the relationship between striatal dopamine and subjective value of cognitive effort. And that subjective value, which we derived from that choice phase of the task, is plotted here on the y-axis, with higher scores representing higher subjective value, so higher willingness to do the task again. They were willing to sacrifice more money in order they were willing to sacrifice less money in order to avoid the task, I should say. And what you can see is that subjective value is a function of task difficulty. So it decreases with more difficult tasks. This just means people are less motivated to repeat more difficult tasks. Motivation is lower for more difficult tasks. But the critical finding here is that the subjective value was also predicted by individual differences in dopamine synthesis capacity, particularly in the caudate nucleus in the dorsomedial stratum, so that people with higher levels of striatal dopamine exhibited higher subjective value, higher willingness to do this task again. And what's important, these same subjects did this task under placebo, but also under methylphenidate and also under sulpiride. And what was interesting was that the subjective value was not only boosted as a function of individual differences, but also by administration of both methylphenidate and sulpiride. And critically, again, as previous, these effects depended on baseline levels of striatal dopamine, with effects being restricted to people with low baseline levels of dopamine, whereas high dopamine subjects, those with high levels of dopamine to start off with, experienced no effect, or if anything, a reduction in value. Now, based on the computational go-no-go model of the basal ganglia proposed by Michael Frank, we can actually also make more specific predictions about the effects of dopamine on this task. Specifically, we predicted that high dopamine states are associated with increased weight on the benefits, here in green, but reduced weight on the costs. And to test that hypothesis, Andrew actually designed a third phase of the experiment. And in this third phase, subjects were again choosing between the high and the low effort task for more or less money. But this time, Andrew sampled choices on either end of each individual point of indifference, which then allowed him to plot the full psychometric curve, which quantifies the degree to which high effort task selection varies with the difference between the subjective value of the high and the low effort tasks. And OPAL model, so this go-no-go model, simulations of that model demonstrated that an increase in sensitivity to the benefits and a decrease in sensitivity to the costs is associated with a steepening of the right side of this psychometric curve, but a flattening of the left side of the psychometric curve, respectively. So changes in sensitivity to the benefits and the costs in this task, so the money versus the difficulty of the task, had this interesting asymmetric effect on the psychometric curve, theoretically and also empirically. So here is the result from the model simulations, but the actual empirical data reproduced that model simulation. So it turned out that dopamine had exactly that asymmetric effect on the psychometric curve, both in terms of individual differences, plotted here on the left bottom panel, with high levels of dopamine being associated with higher sensitivity to the benefits, but lower sensitivity to the costs. And also in terms of the drug effects, with methylphenidate boosting sensitivity to the benefits, to the monetary payoff, but reducing sensitivity to the difficulty of the task. Now that qualitative effect was confirmed by statistical hierarchical regression analyses, but also by dynamic gaze patterns that Andrew measured during choice. And in this third phase of the task, he presented the choices in a slightly different way. And that way he could measure gaze at benefit and cost attributes separately. So he basically said to subjects, do you want the left option? That's the really difficult task, but you get a lot of money for it. Or do you want to get the right option? That's an easy task, but not very much money. And then he presented these attributes separately on the screen and measured eye gaze. He just assessed where people were looking when they made their choices. And those gaze patterns are presented here on this slide, locked to the time of choice. And I'm presenting them separately for in green is gaze at the benefits, and in blue is gaze at the costs. And what you can see, if you just look at the top two panels, is that subjects gaze more at the benefits than at the costs on trials on which they chose the hard task, compared with trials on which they chose the easy task. And critically, that effect, that difference between hard and easy task choices was amplified, boosted by dopamine, both in terms of individual differences measured with PET and in terms of the drug. So what that showed was that the effect of gaze at the benefits versus the costs on choice was greater for high than low dopamine subjects. So that really establishes that the cognitive enhancing effect of methylphenidate indeed reflect, at least in part, increases in striatal dopamine and associated gating of a cognitive task in proportion to its benefits versus costs. And what that set of findings illustrates is that dopamine might well help us to adapt to our environment by arbitrating between different cognitive tasks by altering the weight on their costs versus benefits. Now, I see I've already gone quite a long time, so maybe let me just take a little pause to see if there's questions, and if not, I'll move
Dr.
on.
Coles, this is
Yes.
Mike Friedlander. I'd like to ask you a quick question since you're taking a little break there.
Yeah.
In terms of the baseline dopamine levels and variability between people, is it stable in an individual over time? Do you have longitudinal measures on individuals of that level?
Yeah, very important, of course. We don't in these studies have longitudinal measures, unfortunately. We do have those studies are available, and the test-retest reliability for F-DOPA is high. So there is evidence. So we do have evidence that F-DOPA uptake is relatively stable over time. And so we kind of built on that knowledge here, and we're able to indeed relate drug effects, which were measured on a different session, to dopamine synthesis capacity, which was measured earlier or later.
Okay, thanks.
But yeah, we were in 100 subjects scanned with PET was sort of had ended our budget, I guess.
Okay, there is one other question posted at the moment from Anthony Lamonte. He asked, where do you think the modulation of dopamine during the task is acting? Only at the striatal synapses or at additional sites, cortical, etc.?
Yeah, that's of course an interesting question, and in this particular study we did not measure bold signal as we did in the first study. And in the first study we found that methylphenidate indeed had a selective effect on the striatum. The current study does suggest it acts in the striatum rather than in the prefrontal cortex, based on a number of observations. First, the effects were directly consistent with those predicted from a model of basal ganglia dopamine in the striatum, not in the prefrontal cortex. Second, the effects of methylphenidate were paralleled by those of sulpiride, which is a receptor agent acting on the D2 receptors, which are much more abundant in the striatum than in the prefrontal cortex. And third, the effects were predicted by dopamine levels measured directly in the striatum with F-DOPA PET. So those three observations lead us to think that it's most likely mediated by effects on the striatum, rather and indirectly affecting these cortical representations.
Okay.
Yeah.
All right, thank you. Why don't we hold up another question? Let you get back to your flow of your talk now. Go ahead.
Yeah, I mean, it's a relevant question, and the next bit, the last bit of my talk kind of relates to this issue, but asks about the role of cortex in actually setting the levels of dopamine. Mm-hmm. Indeed, one obvious question is how do we set the value of cognitive work? If striatal dopamine controls arbitration between different tasks, then what controls striatal dopamine? So another way to ask this is how do we set the value of cognitive work, which is not a trivial question, given that that valuation is context dependent. So for example, cognitive work is not so valuable when the environment is uncontrollable, for example, or when it's really volatile, changing a lot, or when it is opportunity costly, right? When the cost of focus is too high of work. And so one intriguing possibility that we will be addressing in future work is that striatal dopamine, which is so involved in cognitive effort, valuation, and exertion, is controlled by top-down signals from cortical regions that compute these key statistical metaparameters like environmental controllability. So more speculatively, perhaps it's the cortex, frontal cortex, perhaps, that prioritizes distinct dopaminergic projections to distinct striatal subregions by setting their baseline tone in an environment-specific manner. And perhaps the detection of environmental controllability boosts dopamine release specifically in those parts of the stratum, dorsomedial stratum, probably, that promotes cognitive work. And indeed, from work with humans that has combined transcranial magnetic stimulation with the dopamine PET, actually raclopride PET measuring dopamine release, we know that stimulation over frontal cortex can alter dopamine release quite selectively in those regions of the stratum that are topographically most strongly connected with the stimulated frontal region. And another line of very relevant evidence comes from work from Arif Hamid and colleagues, which is about to come out in Cell next week, showing that the precise timing of dopamine release in distinct subregions of the stratum, dorsomedial and dorsolateral parts of the stratum, varies as a function of the perceived controllability of the environment, manipulated here by presenting the rodents with an instrumental or a Pavlovian task, in which they have control in the instrumental task, but no control in the Pavlovian task. So specifically, what Arif showed was that dopamine waves starting in a dorsomedial stratum when the environment is controllable in a controllable instrumental task, allowing the animal to boost effortful work, whereas conversely, dopamine waves started in the ventrolateral stratum when the environment was uncontrollable in a Pavlovian task, leading the animal not to work. The point I want to make here is that the striatal dopamine system, which regulates goal-directed effort, is in fact extremely flexible and sensitive to environmental changes. And the importance of this adaptive capacity of the striatal dopamine system is illustrated by the way that our brain copes with stress, something so relevant for our society today. And indeed, disproportionate stress reactivity is generally recognized to contribute to a wide range of brain disorders, including depression, anxiety, etc. Now, the importance of detecting controllability is illustrated beautifully by a video that I'm not going to show because I can't get the sound to work, but it's okay because we're already out of time. But if you find time, then you might look up this monk who makes this point beautifully, that adaptive behavior does not depend simply on the presence of stressor, or mental health does not simply depend on the presence of stressors, but rather on our ability to detect whether they are controllable. So the message is really simple. If a stressor is uncontrollable, then our cognitive response to stress is best attenuated so that we can accept the situation, whereas if a stressor is controllable, then we better fully process it, respond to it so that we can act and exert effort to avoid it. What we need is a meta-level control ability to detect when a situation is controllable, requiring effort, and when a situation is uncontrollable, sorry about this, requiring letting go of effort and control. Indeed, stress-related mental disorders might not reflect simply enhanced stress reactivity, but rather a meta-level failure to compute the controllability of stressors. And so when Romain Liolle was in my lab as a postdoc, now in Portugal, he designed this task to measure subjective controllability. And my sense is that I'm out of time, so I'm not going to go too much detail here. Is that right?
No, you have another 10 minutes or so.
Oh, okay. So then I'm going to just go through this, if people can bear with me the details, because it's I think really interesting work here. He designed an explore and predict task, which allowed him to measure but also manipulate environmental controllability, to the objective controllability of this environment, and then measure the degree to which people then perceived this as controllable. So specifically, human participants, healthy young participants, were invited to explore an abstract environment composed of 3 states: a square and a circle and a triangle, and 3 actions: yellow, blue, and magenta. Just take them as actions for now. And basically, on each trial, participants would see 2 colored stimuli, 2 colored states, and I'll refer to them as states, with the same shape, between which they had to choose. And then on the next trial, 2 new states would be presented. And the shape of these new states was then determined by one of 2 rules: a controllable rule or an uncontrollable rule. Under the controllable rule, the new shape depended on which color was chosen by the participant. Under the uncontrollable rule, the new shape depended only on the shape of the previous states. And these 2 rules, they alternated unpredictably. Subject didn't know they weren't told when which rule was enforced. I hope this makes sense, at least at the abstract level. Yeah. Now, occasionally, after a select number of these exploration trials, which are now here depicted in purple, participants were asked on critical green prediction trials, schematized here, to predict the most likely upcoming states given a state action pair. So concretely, they were shown a colored shape, here this blue circle, and they had to predict what shape would follow. And they would do this for pairs of colored shape, here for both a blue circle and a yellow circle. And so this procedure provided us with an index of subjective controllability, namely, because these 2 counterfactual predictions should only differ in the controllable context, where the upcoming shape depends on the chosen color, whereas they should be the same in the uncontrollable context, where the upcoming shape depends only on the previous shape, which is the same in this case, right? So those prediction trials, by comparing those 2 types of prediction trials, Romain was able to derive whether subjects perceive their world as predictable or controllable or uncontrollable. And in fact, this nice paradigm controlled for predictability and uncertainty, unlike various other paradigms. Now, that index of subjective controllability is plotted here on the y-axis as a function of trial number around rule reversals. So as you can see here in green, focus on the green first, subjective controllability on the y-axis is low when the uncontrollable rule is relevant, but increases with the reversal of this rule into a controllable one. Conversely, in red, we can see that subjective controllability is high when the controllable rule is in place, but then reduces rapidly with the reversal of this rule into an uncontrollable. Now this index of, let's see, what I say here, yeah, the specific pattern of subjective controllability turns out to be better explained by a model that arbitrates between an actor and a spectator strategy based on environmental controllability than a conventional model-based RL algorithm implementing the actor model alone. And here I screwed up something in the slides, but that key, the key model parameter omega that roughly captures estimated environmental controllability was indeed predicted positively by this index of subjective controllability. So this is saying that Romain designed a model that could generate a parameter indexing controllability that we could also retrieve from the behavioral data from these prediction trials. And furthermore, decoding of objective controllability, that by that I mean the rule type, which rule was in place, from the brain data, revealed that a dorsomedial prefrontal cortex region and a dorsolateral prefrontal cortex region were sensitive to environmental controllability. Moreover, decoding performance in the dorsomedial prefrontal cortex also correlated significantly with the impact of that controllability parameter on decision times. And now finally, the final point I want to make is that in a separate experiment, Verena Lee, who actually did a PhD in my group a long time ago, she's currently assistant professor in Leiden, showed that the estimate of environmental controllability measured with this task was influenced by exposure to uncontrollable shocks. In fact, it was reduced. So in this experiment, different groups of participants were exposed to controllable and yoked uncontrollable stress, shocks, using fairly standard procedures. And then after they were exposed to these shocks that were either controllable or uncontrollable, subjects completed this explore and predict task that I just tried to explain, that Romain designed. And intriguingly, the exposure to uncontrollable shocks altered the estimate of environmental controllability, such that participants exposed to uncontrollable stress exhibited greater reliance on the spectator model versus the actor model, and that effect was greatest in those with greater anxiety scores. So it also had some clinical relevance. Now, what we conclude from these experiments, first, is that the human brain computes environmental controllability, and that participants arbitrate between different strategies, control strategies, which we describe here as a spectator or an actor strategy. And they do that based on estimates of environmental controllability. The study also showed that environmental controllability can be decoded from frontal cortex, and that exposure to uncontrollable stress biases participants towards greater reliance on one particular type of strategy, namely the spectator mode of control, because it reduces this estimate of environmental controllability. And so in future work, we want to leverage this line of work, this paradigm, to test this idea that the striatal dopamine, which is so involved in cognitive effort, is controlled by cortical codes or cortical regions that compute controllability. And with that, I want to finish and say that more generally, these data illustrate how dopamine can help adapt the output of our neural systems to our constantly changing environment. Clearly, there's a large number of wide open questions that will have to be addressed in the future. If the striatal dopamine system can switch operational mode, depending on metaparameters of the environment, can we then manipulate the cortex to alter top-down environment adaptive control of subregional dopamine release? And what is the time scale at which this switch can act? Can the switch be flipped according to state changes rather than trait differences? And finally, a question that I know many different groups are interested in as well is, might we conceptualize disordered behavior, mental illness, as representing a normative adaptation to their perception of the environment coded in cortex? And if you want to hear more about these questions, then stay tuned and keep an eye on this website. With that, thank you very much.
Thanks very much, Dr. Coles. We have a few questions, so a few more minutes. Pearl Chu asked, first of all, she said, thanks for a fascinating talk. And she asked you if you could comment on dopamine variability with regard to perceptions of controllability versus actual controllability. So some people with psychopathology like depression would perceive less controllability. So they're wondering how dopamine might be implicated in those situations.
Yeah, so I guess the hypothesis here is that depression, which some forms of depression, which are known to be accompanied by learned helplessness, for example, might be characterized by sort of suboptimal subjective estimates of controllability, and that as a result of that, failure of cortical computation, dopamine is dysregulated. So it's not adequately increased in dorsomedial parts of the striatum that lead us to exert effort. Now, and I'm not sure if this is what you're asking, but there's also work ongoing suggesting that the computation of these metaparameters, the learning of the controllability, is itself a function of dopamine. So there's these hierarchical circuits where dopamine might boost cognitive effort to exert a particular motor response, but it might also boost effort to exert a particular cognitive task, and it might boost effort to exert, it might energize the exertion of a specific strategy or contributing to the learning of this metaparameter itself. Sorry, that wasn't very clear, but there's no evidence for this yet, but I think it's not unlikely that dopamine will contribute to the learning of these metaparameters, although the degree to which these parameters will be plastic will be, I anticipate, a bit less obvious. It'll take a lot more time to learn that the environment is controllable if you've been exposed in early childhood to an environment. Let's put it that way.
Yeah, thank you. Actually, I just want to follow a little bit from that. It's sort of, my question's a little bit embedded in, I think it was your second bullet point. I was just curious about what you think about a sort of a time constant of integration for computing environmental controllability, and whether it goes across situations and tasks, or if it's something that is computed and learned only in the context of the current situation and task, or something we carry with us. I mean, you mentioned learned helplessness, for example, as an extreme example of where you might end up.
Yeah, I mean, the honest answer is I don't know. That we don't know, and the question really concerns not only it concerns we can address that question or think about that question in two different ways. One is, what's the time scale at which the cortex can influence and flip the switch at the level of striatal dopamine? That might be relatively fast, but the complementary question is, what is the time scale at which the dopamine system can reinforce that hierarchically very high level parameter of controllability? And it'll take time. It's an empirical matter, I would say. I really can't address that. I have no evidence to speculate about that, to be honest.
Yeah, understood. Thank you. Anthony Lamontia asked again, he asked, is the representation of controllability assembled in the dorsolateral prefrontal cortex, or is it more distributed, with the dorsolateral prefrontal cortex contributing an executive function, working memory dimension, but other regions regulating other aspects of the information?
Yes, so let's see. Let's see if I can address the question in part by going back to the data. The result of this analysis, of this decoding analysis, suggests that it is to some degree distributed. So we can decode controllability from a network of regions. So that certainly is part of the answer. Then there's a lot of prior knowledge about the specific functions of the medial frontal cortex and how that drives regulatory functions of the dorsolateral functions of the dorsolateral cortex. And so based on that prior knowledge, we might speculate about the specific contributions of these different regions here. Which I would like to abstain from based on these data, but it's well possible that even though we can decode these signals from a distributed network, that they play different roles with the medial frontal cortex, for example, detecting this level of controllability and then transmitting information to the more lateral parts of the cortex that then influence downstream regions. I consider that a possibility for sure.
Okay, thank you. Well, I see we are a bit over time, so just one more last question, and then we'll have to break, I'm afraid, after that. Terry Lorenz said it's a fabulous talk. Thank you. He said, would you speculate a little bit about the role here of other neurotransmitters?
Yeah, no, thanks for leaving the word speculate in here. So I think a really interesting candidate is serotonin. We've done a few years ago quite a bit of, I don't think I have a slide, let me see, on serotonin. No. Some work on serotonin suggesting that serotonin might contribute to this kind of Pavlovian response to suppress behavior in anticipation of aversive outcomes. So that's super relevant to the controllability of stressors, of course. And Quentin Huyssen and Peter Dayan have also already a while ago speculated that this aversive control function of serotonin might extend to the control of thoughts and rumination. Yeah, so that serotonin might contribute not just to the suppression of responses in anticipation of aversive events, but also to the suppression of emotion and thoughts and rumination in anticipation of aversive events. And so so there's that sort of piece of understanding. Then in parallel, there's really interesting work done in the domain of serotonin, showing that the raphe, the dorsal raphe nucleus, the origin of the serotonergic projections, those neurons can also switch operational mode depending on the environment, where certain activation of raphe neurons can promote locomotor activity or suppress locomotor activity depending on threat levels of the environment. So a really intriguing idea is that this mechanism, this top-down control arbitration mechanism that I'm putting forward here, might generalize also to the other systems, in particular the serotonin system, where, for example, in a controllable environment, the raphe is suppressed. Indeed, this is kind of consistent with the work by Stephen Meyer and Amat, showing that the frontal cortex controls the raphe in the context of controllable stress. And that when a situation is uncontrollable, the raphe is released to exert its rumination control function that is so adaptive only if stressors are uncontrollable. There you go. Why wild speculation.
Thank you. Well, even though it's time up, there's one more question. I'll just try to get in quickly if you can. Stephanie Roble asked if the system can be trained, for example, by applying discipline to focusing on challenging cognitive tasks.
It's so interesting, this question of training, particularly in the adult brain. It's very controversial. I think training effects are detectable. Whether they generalize to other situations is the key issue. I'm hopeful for training, cognitive training or cognitive therapy, if it is directed at the right thing. So I would say that if we want to train adaptive behavior, we should expose people to a variety of environments, an environment that requires control and an environment that requires a lack of control, and then teach people how to distinguish between those different situations and recruit the right strategy. I'm interested to see the effects of a training program that is aimed at not just recruiting control, but recruiting control at the right moment and actually dynamically updating that strategy when it is no longer appropriate.
Great. Well, thank you very much. We really appreciate a wonderful talk, Roshan. Thank you for taking the time. Great talk. Thanks, everybody, for attending.
That applause really works. I should introduce that at the Donders.
Some of it's recorded, but some of it's
all of it. No, I realize, but it nevertheless works. You don't feel such an idiot, saying, okay.
Yeah. All right. Well, look, thanks again, Roshan. I think you have another meeting coming up, right? Is that right? At some point, or not? I'm not sure.
I think, well.
Are you done?
Yeah. No, I think there was. Maybe I have to go back to my other Zoom link for
that. I think that's right. So anyway, I won't see you again, but thank you again. That was really a great talk. Really enjoyed it. Appreciate you spending time with us.
Nice to meet you, Mike. Thank you for that.
My pleasure.